Hi, and welcome everyone to this live stream. Today we will talk about tissue microarrays for animal health research. And this is actually a theme which is an evergreen, let's put it like this, because tissue microarrays are not exactly something new as a technique, but it's something that is constantly used and implemented in a lot of different ways, and so it's really important as a researcher working in animal health, being this in a pharma, in a biotech, or in an academic institution, to be able to use the technique, to understand how this works, who are the providers, what are the different aspects that are necessary to consider if you want to use tissue microarrays for your experiments, for your studies. So without further ado, let's start into the discussion. But before, I would say a couple of words about us, about Lab4Pose. Lab4Pose, it's a life science company. We are supporting animal health researchers in pharma, universities, biotech, by providing mainly biospecimens and animal biospecimens from companion animals, cats, dogs, but also horses, pigs, cows. We are on a mission to enable the development of new therapeutics and diagnostics for science, but also reducing the use of animals in the development of human medicaments. So we are enabling a series of different technologies, from organ on a chip to comparative studies in toxicology. So some of the things that we are doing are, for example, we are providing dissociated tumor cells or healthy cells, also from different kind of tissues from these animals. We provide frozen and formalin fixed paraffin embedded tissues for different kind of studies. We provide blood, biofluids, especially used also in diagnostic applications, biomarker studies, and so on and so forth. And then we have a complementary portfolio of ELISA kits, antibodies, cytokines, and growth factors that complement this for the downstream application. So what we are doing today stays in this kind of context. Okay, so what are TMAs? What are tissue microarrays? Tissue microarrays are basically an array, so a series of different blocks, or better said, different cores, different spots that are taken from different samples and set into a mold that is set up to host up to different, 100, 200 different cores. So you can imagine, instead of having one single block per patient, per case, per sample, then you basically have one block on which 200 different samples can fit in. So how is this done? It's basically taking the individual samples. Normally, the starting point is this formalin-fixed paraffin-embedded samples, FFPEs, and basically drilling a sample out with a particular machine. And then these samples are positioned into a mold, which is already presenting this kind of holes, then positioning them inside. This is obviously a quite delicate technique where you have to be quite careful, because we were talking a paraffin mold, so obviously this is heat sensitive, cannot stay around for a long time, because if the paraffin melts, then obviously also the cores would move. And if the cores would move, they touch each other, or they have other kind of issues. And obviously you want to be able to transplant your core in the right spot, so you're able to refine it, and to perform your studies as you should. And so this is a little bit the workflow. Now, normally when you're establishing a tissue microarray, you have different options. So you have the main variable. The main variable on the technical side is the diameter of the core. So the minimum one would be approximately 1 millimeter, and the biggest one would be 2 millimeters, with 1.5 millimeter, like the measure in between, being the measure that it's more frequently used. So if you're just having, a 1 millimeter core, you're probably able to put 200 different cores on the same slide, on the same block. If you go for a 1 millimeter, probably you're a little bit under 100, and if you're 1.5, then you're between, like in this range between 100 and 150 different spots on the different cores on the same block. And this is particularly interesting and particularly important, and we will see this later on, because the choice of the dimension of the core and the number of different cores on the same slide influences a lot the kind of application that you can do, but it also has some implication on the quality, on the TMA, implications on the quality of the data that you can get out of that particular TMA. Now, what are the most common applications for TMAs in general, but obviously also for these kind of samples, for formalin fixed paraffin embedded samples, where most of the time it's target identification and validation. So I have, a particular theory. I want to screen different samples to find out if I am able to find exactly that target in my, for example, pathology of interest, being this a tumor. It can also be another kind of pathology, also an immune related pathology. It can be a cardiovascular related pathology or whatever. So, and normally this is something where I would have tissue available. So something that maybe I would take out of a surgery. Or I've taken because of a transplant, or I've taken because of any other things. Now, transplants are not that common in animals. These are more common in humans, but just to give you an idea of the kind of technology. And so in this case, you want to have many more different kinds of cases, patients on the same slide, and these make screening of your targets much easier, because it shrinks the amount of samples that you have to manage, and this is really valuable. And the other thing, obviously, the other case where you will have to do a lot of screenings is for the biomarkers, and also this is one of the cases. Here I'm talking about diagnostic biomarkers on a tissue level. So probably, this is not the application for like blood biomarkers or biomarkers from biofluids in general, but this is obviously application for tissue-based biomarkers, where you want to understand, you want to grade a particular kind of tissue, you want to understand how does a particular tumor, for example, disease evolve with time or in response to a particular treatment, for example, and you want to understand, okay, how does a healthy versus a diseased tissue look like? What are the kind of downstream applications that you can do with TMAs? Well, on the one side, we have immunohistochemistry and histology, so basic histological stainings. If I want to understand if I have an infiltration, or what's the percentage of fibrotic tissues in my particular slides. These are obviously quite basic things, but sometimes, or most of the times, I want to see the expression of a particular protein, a particular marker inside my slide. And nowadays, actually, many more applications are going in the direction of developing really specific markers also at the RNA level. So RNA in situ hybridization is a technique that is super valuable also for tissue microarrays. And we have a little bit of a hype, but it's a positive one, obviously, in single cell sequencing and transcriptomic analysis. Now, these tissue microarrays will enable you to up your game at a whole new level, because you don't just have a transcriptomic analysis at a single cell level if you want, but also it gives you some spatial resolution. So it gives you the possibility to distinguish inside the cell where are your proteins expressed. Is this near a particular organelle? Is this near the acetylene Golgi? We have in another compartment, where is the mutated RNA or the RNA that I would use as a target. So this is a little bit thing. What are the advantages of TMAs? Well, first of all, it's efficiency. So I have many possible cores on the same slide. So it's much easier to manage this. Practically, I don't have to do 100 different stainings. I can do one, and then I have all the stainings or all the samples stained at once. Then I have consistency. So you can imagine if I have different samples, they all come in different shapes and forms and measures. So it's really, difficult in a way to have a consistent kind of measurement. I will have always to compare different things, and there I have the same diameter, always the same. So also, that's the third kind of advantage. Comparative studies are much easier. So I have all the same. It's really easy to standardize the sample, and so it's much more easy to do this kind of comparative studies and compare apple with apples. It's much more scalable, and so therefore also cost effective. Yeah. If we obviously we have the cost of manufacturing the TMA, but if we compare this to buying all the 150 samples, it's actually much better. And also for the downstream applications, the use of reagents they will have afterwards, antibodies, and so on, I will spare a lot of money. And obviously, last but not least, this is the quality issue during standardization. So if I'm taking the whole block, then I will have regions that are lesions, regions that are not lesions. There are probably regions that will be necrotic, and all the different blocks will have a different percentage of this. If I have a TMA, I can define, okay, I just want the block where I have the lesion. I want a core where I have the lesion. I want a core where I have the normal tissue, a core when I have the fibrotic part. So I can really define the quality at another level. And so this is really important when I'm doing my kind of studies. Now, what are the aspects to consider when designing a TMA? What are the things that I need to keep into consideration and say, okay, when I design it, I will make sure that I consider them in my design. The first one is the one that everyone talks about, but it's actually the less relevant, and it's age. So most of the people, they will say, I just want samples that are, 1 to 2 years old or something like this. This is actually the least relevant, and we basically have to consider, what is it that we are comparing? Because most of the samples are conserved really well inside formalin. So this would not be the issue. And in fact, using even slightly older samples would give us the possibility to, number one, access samples that we would not normally be able to access. Because nowadays, like if we compare the technique now and a technique like 10, 20 years ago, nowadays there's a lot more diseases that are not diagnosed through a biopsy, for example, or just by a sport in the tissue. So it's actually for many diseases we would not have a piece available, and so by using all the samples, we'll be able to expand the amount of samples they have available. And if I actually consider a longer time frame, I'm also able to catch diseases in a more advanced stadium, and also look a little bit of a kind of a time series. I'm looking at a tumor, for example, in the different stadium of its evolution. And this is particularly important because for animals, most of the time, since especially in oncology, we are missing a lot of the early biomarkers. The animals, they get into the clinic and they are already sick. It's already kind of late. Most of the time, the only thing that you can do is really just to you remove the tumor. There's not much more that you can do. So that's a little bit the thing, and we want to be able to detect these as soon as possible, especially if you're developing diagnostics. It's really important to track over time the evolution of a tumor. There are some aspects that we need to consider when setting up a TMA, and these are quality-related issues, and this is especially true if I am comparing different providers. The first one is the length of the cold ischemic phase, so how much time passes between the piece has been removed during the surgery and when the fixation starts. And this is really important. If I leave a piece there 20 minutes on the table, it's different if I put it straight away into formalin. It's completely different if I put it there after 2 hours. And this cold ischemic phase, it's something that we need to keep in consideration, because afterwards you would have necrosis of the tissue. So you would need to keep this in consideration. And then also the timing of the formalin fixation is really important, obviously, because you can overfix the tissue. If you keep it there overnight, it's okay. If you keep it there 2-3 days or whatever, then you might have that it would be more difficult to mask certain antigens, and so also detect them later on in a good way. The other point is storage. Now, if we are talking about storage of the blocks are quite stable per se. There's also blocks that are was stored over 60 years with almost no issue, and they will conserve quite well. If we are storing the slides, there we need to be really, really careful, because sometimes it's really like marker dependent how much I'm able to find, which kind of marker I'm able to find out what is the reactivity. So really important there to preserve the samples in a proper way and proper humidity, but also temperature conditions. And then the last point for the design of TMAs is which kind of criteria parameters do I consider really on the sample side, which kind of samples I put there. And here you can design a TMA in a lot of different ways. You basically have no limit. In fact, you have almost unlimited kind of combination possibilities. But the first point is diversity. You have 150 slots. What do you want to do with that? Which kind of samples do you want to put in this? If you're focusing on a particular kind of tumor, do you want to have different stages of the same tumor? Do you want how many patients or many cases do you want to have there? Which spots do you have? You want to have 1 spot per tumor, 2 spots per tumor, 2 spots from 2 different blocks from the same case. Do you want to have matched pairs? Maybe you want to have tumor, normal radiation tissue. Maybe you also have a lymph node available from the same patient. You want to have also that one. Then quality is obviously another issue. So how well preserved is, are the different pieces? Are you comparing things that are of comparable quality also? What am I putting inside? Am I looking at lesion, non-lesion? Then the layout itself on the grid, this is important also. How accessible are the different spots in there? Is there any particular difference? For example, like it depends a little bit also on the downstream later on. If you're using a machine, if you're using a manual process, if you're using a manual process, you really need to be sure that your reagents, antibodies, they really touch all the spots the same way. So how do you do this? Is there any difference if I have a spot on the side, for example, or in the middle? Hopefully they are, staying the same way, but you need to understand if there's any accessibility issue and also if there's any proximity bias. So if I have two things that are really near, it's easier to assume that they are similar. And it's obviously near also to see some marked differences even when they're not there. So you need to make sure that these kind of biases are not affecting your evaluation afterwards. Then you have an issue with the controls. Which kind of controls do you consider there? Do you have a positive, a negative, but what does it mean positive and negative in your case? Do you know what is the kind of tumor that you're looking, which kind of target that you're looking, or is this something completely new? Are you considering samples also off target from another tissue or from a matched tissue? So these are the kind of considerations. Then you have an issue of the core diameter. We said before, if you're using the 1 millimeter cores, then you have access to much more samples. But in that case, obviously, the quality is a little bit lower because obviously the spot that you take, the kind of core, is much smaller. So it's not that representative of the overall sample. If you're having a bigger core, then the representativeness is much larger, but you have less samples. So you have to consider it too. Annotation. This is another thing. Like you're doing this analysis. Are you doing the analysis the same person doing the evaluation? Who knows about the data? Is the sample blinded? Or are the people doing the evaluation aware of what are the samples? Are they doing the evaluation in a blinded way, or do they know that? And then the grid design, we discussed this before. This has to be suitable also for the downstream analysis. Obviously, if I'm having a particular technique where I might want to have a bad overview of the spatial distribution of a particular marker, maybe it's easier to have a larger core rather than a smaller one. And I want to, depending also on the machine, on whatever I'm using to stain, then later on I want to make sure that my grid fits that. One of the things is, for example, once you have done the TMA and you have cut the slide, so you imagine you have this block with 150 spots, and then you cut the slice and you put the slice on the on your glass slide. Now you don't know anymore what is up, what is down. How do you orient your grid? So it's important to leave some empty spots, for example, at the bottom, so that you have an asymmetrical kind of grid, and then you know, okay, this is up, this is down. So these are all really important parts when designing a TMA. Having said that, I would say we have some time for some questions. Let's wait a little bit in the chat if and see if there's any question coming in. I will check also on the other channels. Okay, I don't see anything for the moment. So having said that, I thank you very much, everyone, for being on this call. If you're looking for support for TMAs, obviously, but also for biosamples, for your animal studies, for your animal research, animal health research, for diagnostics, for therapeutic development, then make sure that you contact us. You can contact us at the different social media channels, just with a DM, or you can contact us at info@lab4pose.com. And I wish every one of you a really nice weekend. Bye-bye.